Hello and welcome to the eOrganic webinar on a novel strategy for soil-borne disease management, Anaerobic Soil Disinfestation, or ASD, presented by Carol Shennan and David Butler. My name is Alice Formiga and I'm the webinar coordinator for eOrganic. eOrganic is the organic agriculture community of practice with e-Extension. We're a community of cooperative extension service personnel, researchers, ag professionals, organic certifiers, and practitioners. You can find information about upcoming webinars as well as articles and videos on organic farming on our website at extension.org/organic_production. This presentation is being recorded and will be posted to our website along with all our many other archived webinars on organic farming topics. We're very excited to have Carol Shennan and David Butler with us online today. Dr. Carol Shennan is a professor of agroecology at the Department of Environmental Studies at the University of California at Santa Cruz. Her research focuses on nutrient cycling and ecological pest management in agroecosystems and sustainability of socio-ecological systems in both developed and developing countries. Dr. David Butler is an assistant professor of organic, sustainable, and alternative crop production in the Plant Sciences Department at the University of Tennessee in Knoxville. His research focuses on developing management practices that increase sustainability of crop production systems in the southeastern USA. During the question and answer period following their 45 presentation, we'll also be joined by Joji Muramoto, also of UC Santa Cruz, Maren Mochizuki of the University of California Cooperative Extension, and Erin Rosskopf of the USDA-ARS. If you have a question at any time during the presentation, you can simply type it into the question box on your screen and hit return. If you don't see the question box, click the small plus sign next to the word question on your control panel to open it up. We'll be reading the questions out loud and we'll answer as many as we have time for in the 30-minute question period following the presentation. Not everyone may get their questions answered because we have a large number of people registered, but we will give you some resources if you have additional questions after the webinar. So now I'm going to hand over the screen control over to our first speaker, Carol Shennan. In just one moment, Carol, you'll be able to just click on the screen to activate the controls.
Okay, did that do it?
Okay, just one moment. Okay.
Okay, thank you, Alice. So as Alice said, I'm Carol Shennan from UC Santa Cruz, and I'm going to talk about the work that we've been doing in California with this novel soil-borne disease management technique, ASD. And we've been working in strawberry production systems.
Muted.
And I'd like to acknowledge Joji and my other cooperators who have been instrumental in doing all this work. So what is ASD? It's a technique that has been developed as an alternative to methyl bromide and other soil fumigants independently in both the Netherlands and in Japan. And it's been found to control a range of soil-borne pathogens and nematodes across quite a number of different crops. And at this point in Japan, it's used by hundreds of farmers in small-scale greenhouse production. This slide shows some of the target pests and crops that have been tested, both in the Netherlands, Japan, Florida, and California. And you can see that it's effective on a range of different soil pathogens and nematodes, and potentially on some weed species, across plants as diverse as onions, tomatoes, and trees like maple and catalpa. So what is ASD? Well, it stands for Anaerobic Soil Disinfestation, and basically there are 3 steps to the process. The first one is to provide an organic carbon source to the soil to feed the soil microbes. And then you create anaerobic conditions, that is the absence of oxygen, by the combination of irrigating the soil to saturation, which removes all the air from the soil pores and replaces it with water, and then covering the soil with a plastic tarp that's impermeable to oxygen. What happens then is that the microbes which thrive in the absence of oxygen under anaerobic conditions, they multiply and feed on the added carbon source that you've put in the soil through the process of anaerobic decomposition. The process results in the accumulation of various chemical compounds that have been shown to be toxic to a range of organisms, which is thought to be at least part of the reason why ASD works. And these are organic acids and other volatile compounds. But other things might be happening as well. there may be some biocontrol happening by the anaerobic microorganisms that flourish under these conditions. We also see a lowering temporarily of pH during ASD that might affect some of the pathogens. And simply the lack of oxygen may be detrimental as well. In all likelihood, it's probably a combination of all of these mechanisms happening. So given that ASD has been successful in a number of systems, we set out to test it as an alternative to methyl bromide for US growers and as an option for organic growers who have disease-infested fields that they need to manage. And we chose to look at strawberries in coastal California and pepper eggplant double crop in southeast Florida as model production systems. And here I'm just going to talk about the California work, and then David will talk about the Florida work. So to adapt the technique to California, we developed it as a bed treatment rather than a flat field treatment. This way it fits readily into current strawberry production and requires less use of plastic than covering the entire field. We also targeted Verticillium dalei as the target pest, and that causes verticillium wilt of strawberries, which is the #1 pathogen affecting strawberry production in California. And we've tested through a series of pod experiments and on-farm trials in a number of different locations in Central California. We've tested different carbon sources for ASD, different amounts of irrigation, and different types of tarping material. And routinely in all of our experiments, we monitor things like soil temperature, something called soil redox potential, which is a measure of how anaerobic the soil becomes, fruit yields, verticillium populations, and then in our most recent field trials, we've started looking at weed biomass as well. So this first slide shows the comparison of the effect of different carbon sources used in the ASD technique. And this was from some pot studies that we did initially. And you can see the tall bar shows the number of verticillium propagules per gram of soil in the control treatment. And then the other bars show the effect of ASD on that number for different carbon sources. Wheat bran and rice bran reduced the number of mycosclerotia from over 40 to about 8. And other carbon sources like ethanol, grape hummus, onion waste, and mustard cake reduced it down to 0 or 1. So this made us very excited and determined to see if we could make this work in the field. So I'm going to talk you through a number of the field trials that we've carried out. The first one, which was done in 2008-2009, was done on a clay soil that had quite high verticillium populations. That's about 20 microsclerosia per gram of soil. And just to give you an idea, you get disease development at about 2. So this was quite a severely infested soil. And we did a very simple experiment, looked at, with a randomized block experiment, looked at a control where we didn't give a carbon source, and the ASD treatment where we applied rice bran at 4.5 tons per acre. For this particular experiment, we used the standard green plastic mulch. We irrigated with a lot of water, more than we actually needed, and as we found in subsequent experiments, we can actually get away with using 3 to 4 inches typically. But the grower wanted to make sure that we got good anaerobic conditions and wanted to apply more water for this particular study. And we ran the ASD treatment for 3 weeks. And this is a picture of the field where you can see the plots where the rice bran has been spread on the surface of the soil. And so what you do is you then incorporate that rice bran, and we incorporate it to a depth of about 15 centimeters or 6 inches. And then you come through and make the strawberry beds. And you can see the white powder is distributed throughout the bed, which is what you want. You want it to be as evenly distributed as possible in the root zone. Then we came in and applied the plastic, as you would do for any typical strawberry production. Only at this point, what we did was apply the irrigation water through drip tape that had been laid before we put the plastic on top. And so this is a little bit of a complicated slide, but it shows, again, the number of microsclerotia of Verticillium dalei. And the blue bars are for the control treatment that didn't receive any carbon source. The red bars are for ASD. And then the green bars that appear in the last two dates is from the neighboring parts of the field where the field had been treated with inline and soil fumigant. So if we look at the baseline numbers, and I'm moving the cursor to show these, you can see that before the experiment started, we had about 20 microsclerotia per gram of soil. After the ASD treatment, that fell to about 14 for the control, but fell all the way down to 3 for the ASD treatment, showing that we got good control immediately after the ASD treatment of the verticillium. Now, months later, during the middle of strawberry harvest, we measured the population again, and you can see that the control treatment had increased up to close to 30, and the ASD treatment had also increased up to about 14, the same as in the rest of the field that had been fumigated. But at the end of the strawberry harvest season in September, the ASD treatment had brought the number of microsclerotia down to about 2 again. So overall, it was effective at suppressing the population through the entire season. So we wanted to repeat this study and see if we could get similar results. So we did it again in 2009 to 2010. This time it was on a slightly lighter soil, a clay loam, and the number of microsclerotia was a little bit lower, 13 per gram of soil. And this time we were compared directly within the experiment to methyl bromide as being the standard treatment. And this time we looked at 3 different ASD treatments. We looked at rice bran at 4.5 tons per acre, at twice that rate at 9 tons per acre, and then a mixture of rice bran and mustard cake. And the reason we used the mustard cake was that it's thought to have some properties for suppressing diseases and suppressing weeds. So we wanted to see if we got better control by mixing in the mustard cake as well as the rice bran. And so here's the data from that experiment. And again, you can see that the ASD treatments reduced the number of surviving verticillium propagules from about 6 in the untreated check down to below 2 for the rice bran at 4 tons per acre, and down to more like 1 or 0 with the rice bran at 9 tons per acre or the rice bran plus mustard cake. And you can see the control was just as effective as the methyl bromide treatment that was included. And we measured, got marketable yields from this experiment, and there actually wasn't much disease development, so there was no difference between any of the treatments, between the untreated check, the ASD treatments, or the methyl bromide control in this particular experiment. So we did another trial at Watsonville, again in a loam soil this time, and we discovered this soil had no native verticillium, so we were not able to collect verticillium suppression data. We included the same 3 ASD treatments in the methyl bromide control, but I wanted to show you this data even though we don't have the verticillium results, because here we got a different yield response. In this case, the untreated check yielded about 75% of the methyl bromide control, and the ASD treatments were intermediate, but the best ASD treatment, which was the rice bran plus mustard cake, only yielded 85% of the methyl bromide yield. So here there was an extra stimulation of yield due to methyl bromide that we can't really explain, because it wasn't related to disease. It's not uncommon, however, that methyl bromide can stimulate yields in ways that we don't quite understand. But all of these, the yields with the ASD were still very economic yields from that trial. So moving on to the next trial, this was done down in Ventura County, which is a warmer area. And since ASD relies on microbes breaking down carbon, they're going to do that more rapidly under higher soil temperatures. So we wanted to see how well this technique worked in warmer soils. This time it was with a clay loam soil that had moderately high native verticillium at 15 microsporosia per gram of soil. And here we compared two different tarps, the standard black tarp and a clear plastic tarp. And one of the reasons we used the clear tarp here was to see if we were getting good weed suppression from the ASD treatment as well. And also the soil temperature would be a bit higher under the, possibly under the clear tarp, and we might get some solarization effect on the weeds. So that was the main plot, was the different types of tarp. And then the other treatments were an untreated check, where we didn't apply carbon or water, an untreated check where we applied water but without any carbon source, and then 3 ASD treatments. Sorry, 2 ASD treatments, one for 3 weeks and one for 6 weeks. And the carbon source for the ASD treatments was rice bran in all cases. And in this case, we applied the rice bran a little differently because we had the equipment that allowed us to incorporate the rice bran within the beds. So the beds were already made, the rice bran was spread on top of the beds and then incorporated directly into the bed. And these are the two kinds of plastic tarps that we used in the experiment, and some of the crew that set it all up. So let me talk you through this data slide. We have to my left, we have the black tarp treatments, and then to the right all the clear tarp treatments. And again, you can see that relative to both controls, the untreated control plus water, the ASD under the black tarps reduced the number of microsporosia by about 80%. Under the clear plastic, the 3-week treatment reduced the number of microsporosia by about 90%. But for some reason, the 6-week treatment didn't do quite as good a job. And we're not quite sure why that would happen. But again, we're getting on overall 80 to 90% control of the verticillium again. Looking at the weeds, though, we counted weeds under the clear plastic mulch, and while there was some reduction with the 2 ASD treatments, there was still quite significant weed growth, suggesting that the ASD treatment is not able to completely suppress weeds, at least under these soil conditions. We did, however, see a growth effect, both from the use of the clear plastic and from the use of ASD. And these 4 photos show you examples of the different treatments. And you can see that the best treatment here was the combination of the clear plastic and the 6-week ASD treatment. And the ASD treatment in the black plastic was also superior to the black untreated control. So if we look at that as a graph, we see 2 things were happening. One was that just using the clear plastic really increased early marketable yields. And using ASD similarly increased early marketable yields. Now, we don't have all of the yield data because we're still taking harvests right now, so we don't know how the final yields will end up. But it does show that using the green plastic, in spite of the weed growth, you could get improved early season yields from the strawberry plants. Okay, so to summarize, from all of these experiments, we've been able to get good verticillium control using ASD, between 80 to 100% in most experiments. We've obtained good yields. In one case, they were equal to both the methyl bromide and the untreated control yields. In another, they were within 15% of the methyl bromide yields. And in the Ventura study, we saw an 80% increase from the untreated control in early marketable yields. And the other thing we found from the experiment I showed and from others that I haven't shown is that standard tarps are just as effective for ASD as the totally impermeable film or the virtually impermeable film. So there's no benefit to spending the extra money on the more expensive tarps. So there are still some questions that remain. One of them is, can we further improve the disease suppression? If we could get it above 90 all the time, that would be great. And one of the questions we have is whether soil temperature is really making a difference, and that if, when the soil temperatures are lower, we need longer tarping periods for the ASD to work. And so we're setting up trials to test that. We've been a little restricted with our ability to do some of the work because we were wanting to be able to add inoculum to the soil so that we could always get verticillium suppression data, but we're having problems with the inoculum test, which has required us to find infested fields for doing all of our trials, which is a little tricky. So we're in the process of testing different inoculation methods as well. We're looking now at asking the question, well, if we use mustard meal, will we get better weed control from using ASD? Or do we need to have high enough soil temperatures to get solarization to occur? And we're also looking at using models to work out the optimum water application strategies for different soil types, so that we could tell growers, if you have this kind of soil, then for ASD to work, you would want to add so many acre inches of water. So we have two trials that I haven't mentioned currently underway to address some of these questions. The first one is in Castroville on a clay loam soil. And here we're testing three different things. We're testing the rice bran versus the rice bran and mustard cake to see which gives us the best suppression. We're doing it at two different dates, once in September and once in October. In this way, the soil temperatures in September will be significantly warmer than during October. And then we're doing two lengths of tarping periods for ASD, either 3 weeks or 6 weeks. And all of these treatments are being compared against an untreated control and a soil fumigant. Then we have another experiment where we're repeating the comparison between rice bran and rice bran plus mustard cake, this time in Watsonville. And here we're comparing it against not only a soil fumigant, but here we're looking at another non-chemical technique, the use of steam for sterilizing the soil and reducing disease pressure. And this is some early data that we've got back from the Castroville experiments, where we were looking at ASD for 3 weeks and 6 weeks, and either with rice bran or rice bran plus mustard cake. And you can see with all of the ASD treatments, be it 3 weeks or 6 weeks, with rice bran or with rice bran and mustard cake, we got almost 100% control of verticillium. So this was great result for us. Maybe we're getting better at doing the treatment in the field, but if we can maintain this sort of result, we'll be extremely happy. And you can see it was as effective or more effective than the soil fumigant that's typically being used. So overall, this technique shows a lot of potential both as an alternative to methyl bromide in strawberry production. But as I mentioned initially, we're also very interested in using this in organic systems, and we have some trials beginning looking at organic fields that have high verticillium populations, because we think this technique would be organically approved and would be available for organic growers. So I think all that's left for me to do is to acknowledge all the farmers who have worked with us doing this work, and the various staff at UC Cooperative Extension, and staff and students from UC Santa Cruz, without whom we couldn't do all of these field trials. So with that, I'll hand it over to David.
Unmuted.
Okay, thanks, Carol. Today I'm going to talk a little bit about some work that I did as a postdoc
Muted.
USDA-ARS in Fort Pierce, Florida, in Erin Rosskopf's lab, and working closely with Nancy Burrell's lab. Just to start off with, I want to give you a brief overview of Florida vegetable production systems. Many of you may not be familiar with that type of system. Most crops are grown in raised beds with plasticulture. As you might expect, the soils are very sandy. Typically, they're grown in a double crop system. In the trial that you will see in a minute, we grew a bell pepper crop late in the fall, and then followed that by an eggplant crop early in the spring the next year on the same plastic. Because of this, there is a summer fallow period in the middle of summer in a lot of these vegetable production systems, which gives us the potential to do some soil solarization. Because it's already a fallow period, it's the hottest, sunniest time of the year, so solarization can work really well and could possibly be integrated with an ASD treatment. We might not see total control with solarization, but if we integrate that with ASD, we might see some better control. The major vegetable crops in Florida that are currently relying on soil fumigants include tomato, pepper, eggplant, and various cucurbits. Some of the key soil-borne pathogens and weeds include Phytophthora blight, caused by Phytophthora Capsule, or Phytophthora blight of pepper, often called, various Fusarium wilts, southern blight, caused by Spluroshimorov Ci, root knot nematodes, Meloidogyne species, especially the southern root knot nematode, Meloidogyne incognita, and also yellow and purple nutsedges are major weed problems in plasticulture vegetable production systems in Florida. The field experiment that I'm going to present to you today was a complete factorial, and this was arranged in a split-split plot. Our main plots were initial irrigation. We had 3 rates of irrigation: no initial irrigation, 2 inches or 4 inches. The subplot was the application of partially composted poultry litter, and I believe in the next slide I have some rates for you on that. And then the subplot would be the application of molasses, either amended or unamended with molasses, as our labile carbon source to start the anaerobic treatment. All of these treatments were solarized during our 3-week treatment period. And also we included untreated and methyl bromide controls. And these were covered with a metallized plastic film, so they are not solarized. So keep that in mind. As I mentioned, our poultry litter application was partially composted. And we applied this in each season to meet the total nitrogen needs for the pepper crop, about 200 pounds of N per acre, estimated to be available to the pepper crop. And this application was concentrated in the center of the raised bed, as you can see in the photos on the right. Our beds were about 3 feet wide, and then we tilled this chicken litter into the top 8 inches of soil. And you can see that in the top photo there after it's been tilled in. The bottom photo you can see immediately after it's been applied. And the reason we were applying the poultry litter not only as a nitrogen source, but more to increase the water holding capacity of these very sandy soils, and hopefully increase the anaerobic condition that we might see during an anaerobic soil disinfestation treatment. Our molasses application was about 4.5 tons per acre. This is on a field scale, would be 4.5 tons per acre, but the application itself is concentrated in the 3-foot bed area, as you can see in the photo on the right. This works out to about 5 gallons of undiluted molasses per 150 square feet of bed area, just to take it down to a smaller scale for you. So it's quite a high rate. This rate has been shown to be successful in some Japanese studies, We haven't really done a lot of work with the various rates, so this is just the rate that we chose for this field study. The molasses was diluted 1-to-1 with water to facilitate application and then sprayed onto the beds, as you can see on the right. The beds were then covered with the clear plastic, so we have the solarization, and irrigated via drip irrigation, those 3 different irrigation rates, 0, 2 inches, or 4 inches of water. Our ASD treatments began in late August, early September. We did this both in 2008 and 2009. We had a 3-week ASD treatment, similar to many of the studies that Carol just presented to you from California. After this 3-week period, we cover the solarization plastic so the soil is not so hot during the production period and to stop that solarization. Then we planted the bell peppers in September. Those are harvested in December, January, and then we follow that by a planting of eggplant on the same plastic in the same beds in February, and then those eggplant are harvested April, May, possibly into early June. We used fertigation in the non-litter treatments to supplement the nitrogen source, since we're not applying the poultry litter in those treatments, and so we can equal out the nutrients that the plants are receiving, so we can see differences depending on our treatments, and then also to supplement the double crop eggplant in cases where the litter is not providing enough nutrients for that eggplant crop. And throughout the seasons, we evaluated yield, plant nutrition, plant growth, and various vigor data. This photo just kind of gives you a representation of the double crop system. We have the bell peppers on the left. These are planted on 12-inch spacing. That's the first crop following the ASD treatment, and then we follow that with the eggplant double crop. As you can see, there are still several holes from the bell pepper crop, and the eggplant are planted down the center of that bed on an 18-inch spacing. We looked at the survival of several plant pathogenic fungi, and to do this we introduced inoculum in nylon mesh packets. We looked at Phytophthora Capsule in both years, and then in the second year we wanted to get some data on some other pathogens, so we looked at Fusarium Oxysporum and Spluroshimorov Ci. Spluroshimorov Ci, as you remember, is the pathogen that causes southern blight. These were introduced at a depth of 6 inches in each of the beds prior to the ASD treatments. Plant parasitic nematodes were evaluated. We did this by rating the crops for root galling and root condition at harvest. And nematodes were also extracted from soil and plant root tissue, counted and identified throughout the experiment. Weeds were assessed immediately after ASD treatments and throughout the cropping cycle. We did this both in the holes where the plants are planted as well as anything that's coming through the plastic, which you might see with nutsedge. Weed biomass was then also determined at the end of each cropping period, the end of the bell pepper and the end of the eggplant in each year. Soil properties were measured as well throughout the treatments, just like Carol showed you for the California studies. So we looked at redox potential as a measure of the anaerobic activity in the soil. We looked at soil temperature as well, as we saw some large differences with solarization, which I believe I'll show in the next slide. And then we also looked at soil pH and various plant nutrients throughout the season, just so we could relate that to what we were seeing with yield. So what did we see in terms of soil temperature with solarization? In this graph, I'm showing the soil temperature at a 6-inch depth, that's 15 centimeters. This graph is actually in degrees Celsius, but I've translated some of those values in the text at the left. Solarization is the small yellow dots, are controls which are under the reflective metal plastic are in the gray triangles, and then the ambient air temperature is in the blue dots. So you can see with solarization we were getting some rather high temperatures at 6-inch depth, up to 115 degrees Fahrenheit during the day. Under our reflective plastics, though, this temperature was less than 90 degrees Fahrenheit, which was similar to our ambient high temperatures. Again, this is in August, early September in South Florida, so a very hot, sunny time of the year. And just as a reference for you, most mesophilic soil organisms, which includes most plant pathogens, have a damage threshold that begins around 102 degrees Fahrenheit. So we were above that, the beginning of that damage threshold. Now some results for our Phytophthora Capsule. What we found was that control of Phytophthora Capsule was equal to that we saw with methyl bromide for all treatments except the untreated check. And this, we've had similar results in greenhouse pot studies. So in any case where we had the solarization or any type of anaerobic soil disinfestation treatment, we've had very good control of Phytophthora Capsule introduced in inoculum. And one of the reasons we looked at Phytophthora Capsule was that there was an initial concern with ASD and pathogens like Phytophthora Capsule that have water-transported spores. However, we did see that ASD is unlikely to increase Phytophthora Capsule incidence based on this high level of mortality we are seeing with introduced inoculum. So that did somewhat allay that concern that was an initial concern we had with ASD. Now moving on to the Fusarium Oxysporum inoculum. I'm going to be showing several graphs now, and they're all arranged in a very similar way. We have the untreated check, which is represented by UTC and the dashed black line typically, and then a methyl bromide control, which is typically in red, also represented by the dashed line. And then our treatments, if the treatment is indicated with a red dagger, that means that treatment differs significantly from the methyl bromide control. And if there is a black asterisk, that means that differs significantly from the untreated check. So in the case of Fusarium Oxysporum, as you see here, we have on the y-axis, Fusarium Oxysporum colony-forming units per gram of soil. And with this, we had actually no impact of applied litter or applied initial irrigation. Our only impact was with molasses application or no molasses application. And in the case of molasses application, control of Fusarium was equal to that we saw with the methyl bromide control, as you can see here. Very good control of Fusarium with applied molasses, regardless of applied level of water. However, all treatments improved control of Fusarium compared to the untreated check, which you see up here at the top and has very high values compared to all of the other treatments in the study. Moving on to Spluroshimorov Control. Our results here were somewhat more variable, and I'll show this as well with a pot study at the end of the presentation. But the germination of introduced Spluroshimorov Control was equal to methyl bromide treatments for all treatments that included molasses or poultry litter. As you can see here, the three treatments, regardless of initial applied irrigation, are all equal to our methyl bromide control. However, at the same time, we also have fairly good control from our untreated check. So there is some variability going on here. As I mentioned, this was only done in 1 year of the study, so take that for maybe as some preliminary data. We also saw some similar results, however, with the applied litter in some microplot studies where we were just looking at litter application and various irrigation rates. And there, actually, even without anaerobic conditions, we were getting control of introduced Spluroshimorov Control with applied litter. As you can see, we were getting about 41% germination with no applied litter and only 5% germination when we had applied litter. With plant parasitic nematodes, we have a different kind of graph. Here you see we did actually have an impact with applied initial irrigation, which is what you may have expected to see with many of the other pathogens. But in this case, the Melodogyn incognita, the southern root knot nematode, populations were reduced by any application of molasses and or litter amendment when initial irrigation of 2 inches or 4 inches was applied. So you see when no initial irrigation, all solarized, regardless of amendment, we had levels of control that were much greater than the methyl bromide control, but with either 2 inches or 4 inches of applied irrigation with amendments of molasses or composted poultry litter, we had control that was equal to our methyl bromide standard. And this was similar to what we saw with weeds. Here, again, with either 2 inches or 4 inches of initial irrigation, weed control in the planting holes, and in the planting holes we mostly saw grasses, crabgrass, goosegrass, was improved by the application of litter or molasses. Again, we can see here much better control than our untreated check, statistically similar control to the methyl bromide standard. And just for a visual representation of that, I have some photos for you. These are both untreated check treatments. You see a lot of grasses growing out of the planting holes during the pepper crop. It almost looks like they're growing in the alleyways, but in fact most of these grasses are growing out of the holes and going down into the alleyways. And this is compared to all of the other ASD treatments. I couldn't name which these are for you, but you can see just visually much better control of weeds in those solarized ASD treatments as compared to the untreated check. Now I'm going to present some results for yield. This is obviously very important, so I've got 2 years of data for you on both the bell pepper yield and the eggplant yield. On the y-axis, we have total marketable bell pepper yield in tons per acre, and then we have our different treatments on the x-axis. In this case, again, we saw no impact of the applied initial irrigation. So I'm only showing you the various treatments, various soil amendment treatments, either no amendment, molasses amendment, litter amendment, or litter and molasses amendment. Starting first with the 2008-2009 season, these would be peppers that would have been planted in 2008 and then harvested in December, January 2008 into 2009. In this case, you can see our methyl bromide control, 2008-2009 is up here, our untreated check, less than that. Here we had greater yields from all of our ASD treatments or any other treatment over the untreated check, but we saw with no amendments and with molasses and with litter only, we're equal to the methyl bromide control. We actually have slightly lower yields with our litter and molasses treatment. And what we think was happening here was just a lack of synchronicity of the mineralized nitrogen from that litter amendment, especially with all of that added carbon from the molasses, just reduced the yields from that initial bell pepper crop. In the second year, however, we saw a different pattern. In this case, with no amendment, which would be the same as solarization only, we actually did not have as good of yields as in the treatments where we either applied molasses, composted poultry litter, or composted poultry litter with molasses. In this case, the yield was similar to the untreated check, and we had greater yields from all of the other treatments compared to the untreated check, and actually with molasses only, our yield actually exceeded the methyl bromide control. The eggplant picture is quite a bit different. We see a similar pattern with starting with the year 2008-2009 where we had much better yields. The methyl bromide check for 2008-2009 is at the right, and the untreated check for 2008-2009. In this case, where we had applied poultry litter, we actually had some greater yields compared to the methyl bromide control. From our data, we don't really think this is a nutrient issue, more of just a soil improvement from the added organic matter of the litter is probably what's happening there. We are supplementing all of these crops with inorganic nutrients if the litter is not applied. So we really don't have big differences in nutrient availability to these crops. In the second year, however, we had a lot of weeds that were coming in from outside of the plot area. So in this case, we had suppressed yields in all cases, even with our methyl bromide controls. Again, we see the same pattern with applied litter. The eggplants actually did very well, and we have similar yields from either solarization alone or molasses amendment. Because in Florida systems we have this summer fallow period, we were thinking that this works really well to use a warm season cover crop as our labile carbon source for anaerobic soil disinfestation. These warm season cover crops can fit well into production systems, provide a variety of ecosystem services to producers. So what we looked at was, in an initial greenhouse pot study, we looked at 2 warm season legumes, cowpea and sun hemp. You can see sun hemp on the right at the top. 2 grasses, pearl millet and sorghum Sudan grass. That's pearl millet at the bottom right. And then we mixed cowpea with each of those grasses. We included molasses and untreated controls. Our molasses control, as you saw, worked very well at controlling pathogens in our field study. And then all of these treatments were irrigated to saturation and covered with plastic. So what did we see? How did these cover crops compare with our molasses control as carbon sources for anaerobic soil disinfestation? In the case of Fusarium Oxysporum inoculum, they performed very well. As you can see, our untreated control here on the right, very high levels of Fusarium Oxysporum. Our molasses control, very good control, and also very good control across all of our cover crops, all equal to the molasses control except for the pearl millet alone. CP plus PM just indicates cowpea plus pearl millet, and the same here, cowpea plus sorghum Sudan. I mentioned with Spluroshimorov Ci, our results have not been as consistent, and this graph just shows that as well. We have our greatest level of control from the molasses, our greatest numerical level of control. Our highest amount of Spluroshimorov Ci surviving comes from the untreated control, as you might expect, and kind of intermediate with many of our ASD treatments. However, we do see this trend, and again I'll emphasize that this is just preliminary data, but we do see this trend with these grass amendments, sorghum Sudan and pearl millet, is actually having some better control than some of the other cover crop amendments as carbon sources. So just to summarize what we saw with the Florida study, we saw comparable yields to the methyl bromide standard, in some cases actually exceeding the methyl bromide standard. We had a high level of weed control from that solarization and the ASD treatments combined, especially when we're talking about grasses and broadleaf weeds emerging from the planting holes. I didn't show any of our nut sedge data, but that was also more variable than the grass and broadleaf weed data. We had promising control of plant pathogens and parasitic nematodes. However, the methods are still being optimized. There's a lot of work to be done yet with rates and timing, length of ASD treatment, etc. ASD, as Carol mentioned, is easily adaptable to organic and conventional production systems. If you can find an amendment that will work well for you, a labile carbon source, it's easily adaptable to current systems, easily adaptable to plasticulture systems. That is one of the limitations of ASD, that it does require the use of plastic, but of course it is highly adaptable to existing plasticulture systems and existing equipment that many farmers have. It is more consistent than solarization alone, but not as consistent as fumigation, as you saw with some of our Scleroshimorov Ci and also weed data. Nutrients, especially in organic systems where it's not as easy to apply nutrients through fertigation, so the nutrients are often applied pre-plant and we're relying on legume cover crops, organic amendments, or soil nitrogen reserves. Managing those nutrients well during that ASD treatment where you're applying a lot of water and there are no actively growing plants is important so that we're not losing nutrients out of the system. The time out of production can be important, although in the grand scheme of things this is a relatively short treatment, 3 weeks to a month. Many fumigants may actually, with coming regulations, may actually require that level of time out of production as well. The technology is in need of optimization to local conditions, systems, inputs, etc. Depending on when the fallow period and planting times are in your region, what inputs you have available. This is all going to depend on what may be the best way to do an ASD treatment in your region. This slide we added just to give you an idea of some of the cost of organic materials and how this might compare to the cost of methylbromide fumigation. Keep in mind that these aren't directly comparable. Our cost of methylbromide fumigation, this is for a commercial application in California, is about $3,000 to $3,500 per acre. Our organic amendments cost just include the cost of the organic amendment. So we're not talking about application cost, management of cover crops if you are using a cover crop source, anything like that, plastic cost. So this is just something to keep in mind when you're looking at these costs. We are working on some overall economic analyses, but we do not have that data yet. As you can see, the rice bran, at the rates that were used in the California studies, the cost per acre would be between about $600 and $1,400. Mustard cake, a little bit more expensive at $1,600 per acre. Molasses at our rate used in Florida would be just over $500 per acre. And then cover crops would, of course, the seeds would be rather inexpensive, although you would have the management costs and also some added opportunity costs just because you have a greater time out of production, although you may have a fallow period that would work well in also. But the cost will vary, of course, but typically we're probably going to be less than methylbromide fumigation, just depending on the carbon source that works well in your area. We have some ongoing work in Florida and in Tennessee, which is where I am currently, examining a lot of pot studies, looking at carbon sources for control of a range of pathogens and weed species. We also have some research farm and on-farm trials looking at vegetables, ornamentals, and strawberry in Florida with various tarping and organic amendments. And then looking at vegetables in Tennessee, tomato and pepper. We have an on-farm study starting next year with a conventional tomato producer in Tennessee. And then we also hope to look at strawberry in the future in Tennessee as well. To quickly give some acknowledgments, this study was partially funded by the USDA Methylbromides Transition Program. Dr. Greg McCollum and Dr. Joe Albano, both at the USDA-ARS in Florida, helped us a lot with some fruit quality and plant nutrition data, which we didn't get into today. I'd like to thank all the people in the Rosskopf lab, in the Burrell lab, also those at the farm who helped a great deal with crop management during this study, and Bill Crawford for designing our irrigation and fertigation system, and then all of those in the Albano lab as well at the USDA. And with that, I believe we'll take any questions that you all have.
Hi, this is Alice again. Thank you, Carol and David, and I'd like to welcome our guests, Joji Muramoto, Erin Rosskopf, and Maren Mochizuki to our webinar, who will be able to speak in just a moment. We're about to start our question and answer period, and for anyone who missed the beginning of the webinar, you can use your question box on your screen to type in the questions and hit return. If you don't see the question box, you can click on the small plus sign next to the word question to open it up. We'll be reading the questions out loud, and we'll answer as many as we have time for. This webinar was recorded, and it will be posted in the coming week on our website under Archived Webinars. And if after the webinar you have additional questions or questions that haven't been answered, you're welcome to use the online Ask an Expert system at extension.org/ask, and you'll get an answer. Finally, we really value your feedback, so we'd very much appreciate it if you could fill out our follow-up survey, which you'll be receiving in an email today. Okay, so this question, to get started here, is for Carol Shennan. It refers to the Salinas trials in 2008 or 9, 2008 to 9, about the Verticillium sclerotia and whether the microsclerotia actually disappeared or whether they were no longer viable.
That they were no longer viable. Okay. We have to try and grow out the sclerosia to see which ones survived. So it's a viability test.
Okay. Is it no problem? Okay. Okay, here's a question also for Carol. Whether the soil temperature needs to reach a critical temperature for this treatment to be effective. And this person is writing from an area that's not such a hot climate, so she was curious how to adapt this technique.
Yeah, we don't exactly have the answer to that yet. We're in the process of doing a series of greenhouse experiments growth chamber experiments where we're looking at a whole range of temperatures to see if we can come up with something like a degree day model. Only it would be degrees EH model to predict whether it would work under different conditions. And also we're looking at whether you need longer tarping periods under cooler conditions. Having said that, the technique has been shown to work in the Netherlands where the soil temperatures are relatively cool. We'll have more specific temperature data in the next few months, we hope, and we'd be happy to send that out.
David, this is Erin. Maybe you could correct me if I'm wrong, but I thought that we had achieved good anaerobicity down to 20 degrees Celsius in our growth chamber studies.
That is correct. I'm not sure what our pathogen data looked like at that temperature, though. But yes, I think we did do a good job of achieving some anaerobic conditions at some lower temperatures. I think we may have even looked at 15 degrees Celsius. Not sure how that relates to control of various pathogens. Just a guess. I would guess that would vary depending on the pathogen. But,
do you have anything to add, Joji?
Yeah, we had the same kind of data. We did test it at 25 degrees Celsius and 15 degrees Celsius, and at both temperature created very strong anaerobic condition. However, Fusarium suppression was not very good at 15 degrees Celsius at all. So it's not just anaerobic condition, but something else is going on.
Okay. Here's a question about different carbon sources. This one came in during Carol's part of the presentation. She said, I assume rice bran and mustard cake are reasonably available in your area. What other carbon sources might be expected to yield similar results for other areas? The closest alternative in this area might be brewer's grain, a byproduct from beer production.
Joji can chime in on this too. I would expect that to work pretty well. I mean, basically, it seems like as long as you have readily available carbon and you put it in at a high enough rate, that the actual source doesn't seem to matter that much. Would you agree, Joji?
Yes, I think so. We tested wine grape pomace, like a squeezed residue, which worked well. So I'm very positive about beer waste.
Okay. Here's a question about how you determine the carbon loading rates used.
Originally, we used the rate used in Japanese study. They used 4.5 tons per acre as a standard the metric, like 10 tons per hectare. So that was beginning standard, and then but we're doing bed treatment instead of flat treatment, and which seems to require more carbon sources. So we ended up doubling the rate for rice bran, like 9 tons per acre at this point.
Okay, here's a question about irrigation rates. This one I think is for David. He had talked about different levels of initial irrigation, and a listener was wondering whether you applied that rate of water a single time or whether you also maintained that level of water.
No, that was applied a single time. So we irrigate, we begin the study, all our amendments are incorporated, they're covered with plastic, and we have the drip irrigation out there. And then we start to apply the water. We want to apply 2 inches in one treatment, 4 inches in the other. That's a one-time thing. It takes several hours. And then after that, irrigation is the same across the study. So it's just a one-time treatment, but it does take several hours to put that much water out through the drip irrigation, of course.
Okay, let's see. How would you evaluate the impact on soil health and nutrient availability on organic systems? You suggested, let's see, I'm not sure I understand this. You suggested, but how do you do well managed?
I assume that's talking about the nutrients. You know, I think we have some data on that. A lot of it still needs to be analyzed. But I think it's just keeping in mind that as we look through these studies and try to optimize ASD to various conditions, if we're not careful about what kind of amendments we're applying and the rates we're using and the rates of water that we're using, we could be losing a lot of nitrogen through that system, especially nitrogen, but also some other nutrients as well. I don't know if Carol wants to add something to that from their studies as well, maybe.
Well, that's something that we're wanting to look at more. But I think part of it is retaining a good high amount of carbon in the system so that you don't lose too much nitrogen. Joji, do we have any data that we can talk about?
We haven't really summarized yet, but yeah, definitely nitrogen is one of the important part of it. We should be careful not to lose a lot of nitrate to leaching. Also, denitrification during anaerobic process is another concern. So we are trying to monitor that in the future study.
This is kind of a related question. Could you elaborate on protecting against nitrate and other nutrient leaching during ASD while still having nutrients available for the following crop?
I'll go ahead and take that. There is going to be a lot of available nitrogen just because of all that mineralization that's going on during your ASD treatment. And if you're not continually applying irrigation, you're not going to have leaching of that nitrate. You may have some denitrification depending on how anaerobic it is. Probably that's going to be limited because it is a tarp system at the same time, so you don't have a lot of atmospheric exchange. So I think just arriving at that rate of irrigation that works well for you, but not going above that rate that works well for a good anaerobic treatment. And then also, as just to reemphasize what Carol said about making sure that there's enough carbon in the system to try to bind a lot of that nitrogen up into organic compounds rather than it being easily lost into the environment.
I think one of the keys, this is Erin, one of the keys is to make sure that you look at your irrigation rate so that you are achieving saturation, but you're not flooding the system.
Yeah.
You don't want to see that water running out of your beds.
Yeah, good point, Erin.
Okay, we have a couple more questions about different carbon sources. One person was wondering what the source of the molasses used is.
It's feedstock
that
molasses?
was waste molasses, just a byproduct of the sugarcane processing in South Florida. I assume that's from U.S. Sugar, I believe. But I'm also doing some studies now in Tennessee using feed-grade molasses, which is sold readily for livestock feed, and it's fairly inexpensive. I think that would work well for an ASD treatment as well.
Okay, great. Another question about carbon sources. What kind of organic matter would be locally available and appropriate for solanaceous crops in the northeast U.S.?
That's a good question.
All right. Well,
Northeast.
if we can maybe post some additional resources about that on the webinar recording page,
Yeah,
if we can find
I'd have to
some
do some
afterwards,
research on that.
we can try to contact the person afterwards. Let's see. I'll go on at this point. I'm not sure I understand this question completely, but hopefully you will. No one spoke about the EH readings. Could someone comment on those readings in both presentations?
Yeah, we didn't present all the EH data that we have. What we found from pod experiments is that we got very good verticillium suppression data if we accumulated 50,000 millivolt hours below 200 millivolts. So basically, that's saying that we need to have a significant period of time when the EH is below 200 millivolts for the ASD to work on disease suppression. Does that answer the question?
Hope so.
And we achieved those levels in most all of our field experiments.
Okay, yeah, the questioner can retype in another if that hasn't answered your question. Let's see, here's a question for David. Have you tried ASD in a soil called Chrome type in Miami-Dade County of Florida?
We have not, and actually I'm sorry, I lost my sound for just a minute or two, so I'm sorry if I missed anything, any questions to me, but I am back now. We have not tried that.
Okay. Let's see, here's another question. How does this treatment impact the soil life or biology within the soil, which organic systems strive to maintain?
That's a very good question, and we can't exactly give an answer except that once you punch holes in the plastic, the system becomes aerobic again very quickly, and whether you get exactly the same microbial flora and fauna develop after the ASD as was present before, we haven't had the money and wherewithal to test that yet. I mean, the one thing we can say is that soils flood periodically naturally, and so the soil biota is to some extent adapted to that. And it does seem that there's no, we haven't seen any negative effects on plant growth following ASD. But we don't actually have soil microbial population data that we can point to. Erin, do you want to add anything to that? Okay,
sure. We do have a little bit of information for the Florida system, and what we have been able to see is that there's a pretty high increase in bacterial populations anytime molasses is added to a system, regardless of whether or not it's in the ASD or any other type of system, and generally a decrease in fungal colony-forming units that we're able to plate for plate readings. We do see an increase in the trichoderma population in ASD treatments as well. And we hope to be able to, like Carol said, get some additional funding to look at the more specific changes as we have collected total DNA from all of our ASD programs. We just haven't had the opportunity to look at those sequences.
Okay, here's
the
another
priority
question.
was to see if the technique worked first before we went into the looking at the detailed sort of ecology of the system.
Okay, here's another carbon source question. Can straws of grains work in this technique?
This is David. I would think that tree, Joji, you might want to chime in then as well, but I would think the carbon is just not available enough for the microbes during that time. That's going to mostly be very recalcitrant carbon compounds, and it's just not going to break down very quickly enough to create strongly reducing conditions in the soil.
Yeah, this is Joji, and I agree with David. Japanese studies showed the same result. They tested rice straw, and it didn't work very well.
Okay. Another question, do you have any plans for long-term studies on the same infested sites? Do you have any ideas on how long it will take the pathogens to adapt or what to do to limit pathogen adaptation?
We are actually trying to get money to look at sort of realistic rotations to see, partly to get at that question, to see after you've used ASD how long you could plant verticillium-sensitive crops, for example, and to look at using ASD in conjunction with other techniques like growing broccoli, and because we know broccoli residue is somewhat suppressive to verticillium. So we're hoping to get money to do some longer-term studies, yes.
Okay.
I would hypothesize, though, that it's less likely to show pathogen resurgence in a system that's as complex as ASD is. You know, rather than like a fumigant creating a microbial vacuum, you're actually really increasing microbial activity.
There has been some work in the Netherlands with their perennial system, however, not an annual system, where they went back and tested the soil after 3 years of ASD and it was still suppressive. But that hadn't, of course, been disturbed with tillage.
Okay, here's another question about EH levels. How do we know we've achieved those EH levels to achieve fusarium suppression on the farm? Do we need special measuring equipment, or is there a rule of thumb we can use?
Do you want to take that, David?
That is a good question. You know, I really don't know just what you could do on the farm. You will, if you have anaerobic conditions, you will be able to smell it, actually, at some point. So that may be your best indicator, just, you know, something that you don't have to spend any money to do. Joji, do you have any other further thoughts on that?
Yes, I recently learned from Japanese colleagues. They use the test strip for Fe2+ ion, which is formed under anaerobic condition. We need to do more studies this, but they can semi-quantitatively test the Fe2+ content in soil solution once you extract. So that might be in the future correlated with the EH level, and we might be able to use that simple test in the field. But and still, research needs to be done.
Okay, how would high soil organic matter, 5 to 7%, affect ASD?
I would think that that would be a benefit.
Yeah. I was going to say the same thing. I think if you've got good active soil microbial populations, I think it would be a benefit.
And that helps increase the water holding capacity too. So
yes.
Right.
Okay, it appears that solarization is a critical aspect for a successful ASD disease and weed suppression. Given the need for solarization, there's another question about temperature, what the minimum temperature required is.
From our data,
okay,
go ahead, Erin.
I do feel like in the Florida system we get extra weed control benefit as a result of that increased temperature, but I would say that it's the microbial activity that's really far more important, because we know that that solarization suppression of weeds does not last through the season.
Yeah, and I would just add to what Erin said that I don't know that the solarization is completely necessary for the ASD treatment in Florida. The way we did it in the field, we only had ASD treatments that were solarized. In the greenhouse and in growth chamber studies, though, we did have good pathogen suppression without solarization, and I think Carol showed that with her data as well. So I wouldn't say that the solarization is necessary, but it can definitely augment any treatment effect you get with ASD.
Yeah, it may be more necessary for weed control, but not for pathogen control.
Right.
Okay. Because crucifers have herbicidal and antimicrobial properties, would the use of mustard cake as a carbon source have an effect on other plants used in the crop rotation?
That's a good question, and we're actually doing some tests using different rates of mustard cake in a separate study on an organic farm nearby to look at the effect of mustard cake on weed suppression and on crop growth. So that is a possible effect, but we'll have some answers in a few months.
Okay, we have time for one final question, and that is someone was wondering how the system was tested for ornamentals and what plant species and what, let's see, what did you want to control or improve? Do you know of any studies?
Erin, do you want to take that?
Sure, we're actually just starting our ornamental studies, and for the most part, what crop you plant back into it is not as important as getting good ASD treatments. But that work is what we're just starting now.
Okay. All right, well, we're running out of time, but I'd like to thank you all for your questions. And as I mentioned before, if you have additional questions, you can use the Ask an Expert system at extension.org/ask. This webinar will be posted on the eOrganic website at extension.org/organic_production within the coming week under archived webinars. Thank you so much for coming again today, Carol, David, Joji, Erin, and Maren, and thank you all for joining us.
Yes, thank you, everyone.
Thank you.
Thank you, Alice.